Welcome everyone to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Phenotypic Screening Workflows to Better Characterize Brain Disease. I'm Jeff Bugaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. High content analysis is becoming increasingly important for the drug discovery process. The development of new phenotypic screening assays and equipment that can generate significant amounts of high-quality imaging data affords investigators the ability to follow individual biomolecules within cells, allowing them to observe their effect on metabolic pathways and cellular structure. Many organizations have put significant time and effort into developing high-content imaging platforms that provide researchers the ability to not only quantify the information contained in the images, but also to provide solutions for data management and data analysis following image processing. Let's meet our panelists for today's webinar who will discuss their current phenotypic screening methods and describe their experience using high-content screening systems and software from PerkinElmer. Our first speaker today is Satish Katembe, Assistant Professor of Stem Cell Biology in the Division of Microbiology, Tumor, and Cell Biology at the Karolinska Institute. Dr. Katembe will provide insight into his newly developed cell morphology-based screens to identify small molecules affecting glioblastoma multiforme, the most aggressive form of brain cancer. The second presentation will be given by Daniel Heppner, investigator at the Lieber Institute for Brain Development. Dr. Heppner will discuss his work on phenotypic assays of biochemical pathways associated with schizophrenia and the development of a high-content assay for polygenic neurodevelopment disorders. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentation. We will try and answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. So, without any further ado, let's begin our webinar. Satish, the floor is now yours.
Thank you, Jeffrey. I'm Satish Katembe from Karolinska Institute in Sweden, and today I will present two research programs we conducted here using image-based screening. Both of these research exercises were done using operator-based screen. The first one was done on stem-like cells isolated from glioblastoma multiforme brain tumors, and the second was done on embryonic stem cells, both using the operator instrument we have at the Institute. So if you look at drug discovery, one can broadly classify it as a target-based drug discovery or a phenotypic drug discovery platform. And phenotypic drug discovery is not a new concept, but it has recent improvements with high-content imaging systems for producing high-quality images have aided or have allowed for this approach to be effectively used. So this is a project that we performed on stem-like cells isolated from human glioblastoma multiforme tumors. Glioblastoma is one of the most devastating diseases with a very short median lifespan of around 15 months, even with chemotherapy and radiation. So the aim of this project was to identify cellular phenotypes that are enriched in these stem-like cells from glioblastoma multiforme, and that these features are not necessarily associated with its oncogenic transformation. So the whole aim was, if we find such features, so this would be an acquired feature because the cell is cancerous and stem-like, this feature gets enriched, and if we find such a feature, this can be used as a very good target for developing new therapies against these devastating diseases. So this is what we went about doing with the different cancer stem-like lines from these tumors, and we performed a phenotypic-based screen. Basically, it was to expose cells with different compounds and look how the whole cell morphology changes So this was done by using Operata's Z-stacking feature, where we could take optical sections at different level on the cell, and we counter-screened these compounds on embryonic stem cells, fibroblasts. We also developed co-culture and different other cancer lines. And we identified a few compounds that seems to be specifically killing these stem-like cells from glioblastoma multiforme patients. So we identified 17 such compounds, and this 17 compounds were again pushed into a pipeline on in vivo xenograft model using zebrafish, which then identified few other 3 compounds that could eliminate transplanted cells from glioblastoma multiforme. And this were further evaluated by various cell-based assays where we evaluated cell cycle, toxicity, efficacy, effect on mitochondria, apoptosis, autophagy, and so on and so forth. We then turned to image-based analysis of these effects using Operata to see what exactly happens during the course of compound addition and after a while after the cell is exposed to compound. And what we saw was one compound, vacunol-1, which was the most potent in our screen, caused cells to accumulate lots of vacuoles in them. So this compound caused these cells to start accumulating vacuole uncontrollably. And over the time period, cells started to burst because it could not hold so many vacuoles inside. As you will be seeing in the following movie, it's a time-lapse movie based using Operata screen, where we see that cells, after getting exposed to compound, it starts to accumulate these vacuoles. And the vacuoles keep accumulating, and it keeps accumulating, and cells, they lose attachment, they round up, and then they burst. So if you look at the center of the movie, you can see one cell actually rounding up and bursting. So when you actually look very closely, we could see that cells were getting vacuoles were getting accumulated inside the cell. So we wanted to know how these vacuoles were coming. Are the cells taking in liquid from outside and collecting it inside, or was it some internal process? Just to evaluate this, we added a dye called Lucifer yellow. And we could see that cells that were exposed to vacunol started to accumulate Lucifer yellow inside the cell. As you can see in this panel B and C, when compared to DMSO, you can see more green dots. And the zoomed up version of these cells can be seen in D and E. So the same imaging system could allow us to zoom in, zoom out, making the whole screening process faster. And we also could see that these dye that were used were actually getting accumulated inside the cell. Now we also did, since we did a Z profiling, Z stacking of imaging, we also could see what happens exactly on the surface of the cell when the compound is added. And that you can see in panel G. When a cell is exposed to vacunol, it immediately undergoes a process called ruffling. And once it ruffles, it starts to contract itself, and it rounds up, it starts to bleb, and finally it bursts. So what happens outside of the cell and what happens inside could be easily recorded by this high-content screen. So this project led us to identify a compound which targets very specifically the cancer stem cell isolated from different patients. And it targets them and stimulates them to undergo a process called macropinocytosis, which is active actomyosin dynamic, regulated by actomyosin dynamics, where cell ruffles, Then it kind of goes out, protrudes, and belches some liquid and starts collecting them as vacuoles inside. It is not having time to process these vacuoles, and so it loses its attachment. It becomes round. There's a heavy loss of ATP, and then finally the cells give in and they burst. They seem to be very highly enriched. So what you see here is how an image-based phenotype profiling approach using a system that we have here could identify unique properties or acquired vulnerabilities of cancer cells which have stem-like features isolated from these patients. So this is one exercise we did. The second exercise was on embryonic stem cells. So we wanted to use a similar phenotype-based profiling approach to identify some phenotype with which cells can be fine-tuned and its proliferation studied much better. So we used mouse embryonic stem cells as a platform here, and we performed a same image-based screening after addition of different compounds. And what we see here, we measured colony morphology here. So when compared to control, we saw different phenotypes. Compound produces different phenotypes. They either produce cells, they kill the cells so there was no proliferation, or they partially induce proliferation, or they induce proliferation much better than the control. So the resulting colony size were much bigger. So that is represented as phenotype 4. So we also did a parallel screen using zebrafish and human embryonic stem cell. So human embryonic stem cell also confirmed these 87 compounds that increased colony size in mouse. And when we rescreened the entire library on zebrafish using GFP as quantification. So the zebrafish used here for screening were transgenic, expressing GFP under islet 1 promoter. So basically all cranial neurons glowed green, and we did a screen where we took bright field and GFP images. So the bright field imaging helped us identify compounds producing different developmental toxicity like cardiac edema or developmental delays or developmentally lethal. And the GFP was then, as shown on the right side of the slide, was quantified. And this process identified a compound called D1, which increased or which produced the fish to produce more GFP. And it also increased, it also had the same effect as on embryonic stem cells, both human and mouse, where it increased their colony size. So this is how a transgenic zebrafish, islet 1 transgenic zebrafish looks like. So the top panel is the lateral view, where you can see the parts of the brain and the spinal cord glowing green. And the parts of the brain that is glowing green is actually zoomed up, and the bottom panel shows the dorsal view of that region. And you can see all the cranial neurons numbered here in Roman numerals displayed. And this was used as a readout, and to examine further on the effects of D1 on these cranial neuron development, we exposed the fish again and took better images. And that is what you'll see in the next slide in the cranial neurons. And we could see not only the brightness, the GFP intensity of GFP was higher. We could also see the number of neurons at day 2 as well as day 3 go up in these fishes that were exposed. So we went back to our phenotype-based screen just to understand what is going on, and we kind of used mouse embryonic stem cell as model here, and exposure to D1 indeed produced more larger colony size. So the number of colonies as shown as sphere counts were more. We could also count, when we performed cell counts, we also saw an increase in cell count. And when we did a dilution series with these compounds, we could see that the compound produced increase in proliferation at a lower concentration. And as you increase the concentration, it was toxic. And this was done both as an image-based characterization as well as quantifying ATP. So the percent viability graph that you see is also a reflection of the image-based analysis that we did. So the concentration that produced larger colony when pulsed with EDU also increased in EDU accumulation, indicating a more number of cells were in S phase. So similar image-based quantification was also done for human embryonic stem cells, where you could see compound D1 added at lower concentration produced this massive expansion of human embryonic stem cell colony when compared to higher concentration, and that is reflected not only by the colony morphology, the phenotype, but also on the cell counts and ATP that were measured at day 4 and day 10, indicating that this such phenotype-based profiling can be very, very profitable if used in a context of with multiple models. So we wanted to understand how this whole increase in proliferation is happening. So first we wanted to see whether it affects pluripotency of the cell. So what you see here is we have identified one compound that increases ESL proliferation, both human and mouse. The same compound also has an effect in vivo in zebrafish, transgenic zebrafish, where it shows an increase in motor neurons. So the question was, does it compromise the pluripotency of embryonic stem cells? And does it have an effect on apoptosis per se? Does it suppress apoptosis, and that's why you see an increase? And we saw no such effect on qPCR, immunostaining, and even differentiation did not show us any effect on pluripotency. And we also saw no effect on apoptosis. So when cells were exposed to this compound and we stained for active caspase or did an annexin-5 FACT assay or calcium response, we could not see an effect, indicating this compound was not really affecting apoptosis. So cells that had to die were dying. And we also did a whole cell patch clamp to see there was no effect on any ions getting in or out of the cell. So we went back to our image-based analysis and we did a time-lapse recording of these cells exposed to the compound. And then what we saw was that this compound stopped cell from blebbing. So it was making the cell more rigid. So this is shown by the graph. The time-lapse analysis, microscopy analysis is shown by the graph that we plotted. So the red one is the cells that are exposed to the compound. And you can see that they really don't bleb a lot when compared to the black line graph that reflects the control DMSO-treated cells. So when this was coupled with a microfluidic-based imaging and FACS analysis, we could see that indeed there was no change in cell volume, but the cells appeared more rigid. So this was confirmed by using atomic force microscopy. And we indeed saw that the cells were more rigid. So we had to apply more pressure to actually press the cell when compared to the DMSO treated cells. So this indicated that the cells were rigid, and that was an immediate effect, and that was kind of having an effect on its proliferation. So we have analyzed various markers, and we identified cortactin as a marker that actually moves from the into the cell periphery when cells are exposed to T1. And this causes more, cortactin is a nucleating protein, so it causes more rigidity by making more actin filaments, and the cell becomes more rigid. So we have also identified another target called nucleolin, that also goes, the localization of nucleolin goes higher inside the nucleus that you can see in the right side panel when compared to DMSO treated, indicating that nucleolin is also one of the proteins that are affected upon D1 treatment. We have also done transcriptomics on these cells, and we identified many proteins and different pathways that go up or down. However, further analysis has to be done on this to confirm. So we wanted to see if the same effect can be produced in vivo in mouse, and so we performed an in utero. So this is an in utero injection experiment with D1, and what you can see, this is a diagrammatical representation of how the injection was carried out. So a pregnant female was opened up, and embryos were injected with D1. Then we put it back, sewed it up, waited for 3 days, and examined the ventricular region of the cortex. And what we saw, panels represented by 1, 2, 3, 4. So 3 and 4 are D1 treated, and 1 and 2 are DMSO treated. So what we saw was an increase in EDU positive cells in the ventricular region of the developing cortex, and D1 injection did not cause any lethal defects to the embryo. That is seen at the top right hand side corner. So embryos look normal, and there was no effect on the brain or the body weight. So this gave us an idea that this actually works in vivo. So such a phenotype based screen can actually be quickly evaluated in vivo. And we also used another stem cell system called hematopoietic stem cell system in mice, where we injected D1 into mice, and we evaluated using FACS the amount of hematopoietic stem cells that are enriched in D1 treated mice. And indeed we saw when mice are injected with D1, there is an increase in hematopoietic stem cell system. So these two research exercise that I have described actually originates from a phenotype-based profiling approach. So the first one on cancer stem cells and the second one on embryonic stem cells. They have been simply done on using an image-based screening approach, and that has resulted in identification of 2 different compounds that produces an effect, one on stem-like cells from tumors, another on mouse embryonic stem cells and human embryonic stem cells, with 2 different effects, indicating that different cells have different unique features, and that can be very easily accessed by performing a simple image-based analysis. So with that, I would like to end my talk here, and I would like to acknowledge my team members and collaborators at Karolinska Institute and Uppsala University, and also 2 other companies, Glionova AB and K-Life Solutions, that also participated in this program. With that, I would like to thank you.
Thanks, Satish. That was a great presentation to get us started, and you provided us with some great insight into how high-content analysis can be utilized as a small molecule screening tool for the drug discovery process. Before we move on to Dan's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. So let's get our last presentation underway. Dan, you have our attention.
Hello, this is Dan Hepner. I will tell a scientific story about early events and specification of the nervous system from pluripotent stem cells. I'll not focus on our scientific results, but rather on how we approach problems in psychiatric disorders using concepts from developmental biology and tools from high-content analysis. The outline of my presentation will be first to introduce the problem of heterogeneity, to provide a solution based on our understanding of developmental biology, and end by describing a high-content assay that we've developed using these concepts. The terms in this slide are predominantly clinical symptoms of schizophrenia that appear first in late adolescence, but are widely believed to be manifestations of much earlier developmental defects. The problem of genetic heterogeneity in psychiatric disorders. Neurodevelopmental disorders such as schizophrenia are common. They affect roughly 1% of the population, of all populations worldwide. They have a high heritability. Genetic studies reveal that many common risk variants exist, but each has a small effect size. There are rare variants such as CNVs that have much higher penetrance, but often include many genes. There's genetic heterogeneity as a key factor in these disorders. One is that common risk genes are associated with diverse phenotypes. So many people can have the same variant, but show quite different clinical phenotypes. But even with CNVs, these multi-loci events, the phenotype is highly variable. The Manhattan plot on the right is a representation of chromosomal position on the y-axis and probability of risk association on the y-axis. Red bars have achieved genome-wide significance. This is the current state of the art in psychiatric genetics. So unlike disorders with a single gene of major effect, schizophrenia is a disorder that has multiple sources, and it's really not clear what gene to focus on from this long list. So our response is to focus on pathways that show convergent evidence of risk association. O'Driscoll et al. Produced a pathway association map based on candidate genes in the loci from the previous Manhattan plot. ERB3 turns out to be a focus of many convergent pathways in this diagram. Alzheimer's disease, Fragile X disease, excitotoxic cell death, and schizophrenia are all associated with signaling through ERB family receptors and downstream PI3 kinase. Variation in the ligand itself in the bottom right is also seen with genetic risk. Work from our lab and others has established a signaling model that balances neuregulin signaling with BMP activity in induced pluripotent cells and in neural stem cells. These pathways have developmental consequences in pluripotent cells, where they regulate a switch between pluripotency and body formation. In the top right, activation through neuregulin leads to elevation of NANOG, the pluripotency factor, and repression of MYC-L1, a transcription factor associated with body formation. In the bottom, this reference demonstrates that later in development, neuregulin signaling has important functions in the brain, both in behavior and in synapse formation. Our experimental system involves embryonic stem cells, or induced pluripotent cells, grown as a monolayer on matrigel. In the top left shows that we plate cells in the presence of the ROCK inhibitor, Rho kinase inhibitor, for 1 day after passage, and that promotes survival. After 1 day, we remove ROCK inhibitor and grow them for an additional 5 days before passaging, and that's these cells can be maintained in the pluripotent state for many, many passages under these conditions. What we noticed during time and culture, that is after passage, that there's a reorganization of a transcriptional hierarchy. So in the top row, what we're showing here is a time course, a 4-day time course, where one sees red stained indicating OCT4 levels, and just below that, green stain indicating NANOG levels. With time, the signal, the red signal and the green signal, becomes more and more associated with colony edges. So the edge in day 1 and day 2 is seen by the large black holes, and on day 3 by the pinhole that's surrounded by red and green. Below in the scatter plot, we're plotting the relative signals from OCT4 in the x-axis and from nanog in the y-axis. And although the relative signals through time in the colony don't change that much, the cells that have high levels do. So in the third row, I'm representing the same cells that you see in the top two rows as in XY position, and they're colored based on distance from the colony edge, where red is closest to the edge, orange is a little further away, etc. And if we superimpose those distance map measurements onto the OCT4 nanog scatter plot, it's clear that, as indicated by the red arrow, that the highest OCT4 nanog, the most pluripotent cells, are associated with the edge, the colony edge. And as one goes further and further away from that edge, as we see in row 3, the gradient goes from red to green. Likewise, in the scatter plot, the gradient goes from red to green in the high nanog OCT4 population to the low population in the bottom left. By qPCR analysis, we see reduced abundance of OCT4 and nanog through time. In the three red arrows indicate that on day 1, 2, 3, 4 in culture, there's a reduction in the total levels of OCT4 and POW5F1 or and nanog message. At the same time, there's an elevation in the right of pro-neurogenic transcription factors, SOX21, SOX2, and SOX3, as shown by the blue arrows. Interestingly, the vertical dotted line indicates cell passage. So if we look on the left, in the OCT4 nanog plot, levels get to a low point. After passage, they elevate up to the same baseline levels and then drop again in a cyclic pattern. Similarly with the proneurogenic factors. So what this indicates is that these cells are, although they've begun to show lineage priming towards the neural lineage, they can be reset to a primitive state simply by passage. So as we culture cells, as we culture pluripotent cells, we're actually kind of watching development, even though we think we're growing cells in the pluripotent state. They are showing evidence of lineage priming, but that's an unstable state that can be reset. This system is sensitive to proneural induction and also to promesodermal induction. In the bottom, I'm showing you control cells that have been grown for 3 days in the presence of control conditions, or below that in the presence of a proneural environment that contains noggin and NSB. And if you compare the top panels to the lower panels, it's completely clear that there's an elevation of SOX21 in green, not at the colony edge, but away from the colony edge. Likewise with SOX2. The scatter plot in the center that shows showing each of these transcription factors as a different color makes it completely clear that there's a significant elevation of SOX2 and SOX21 below when cells are treated with proneural factors, and that's not true in the control. So this is how we are quantifying these types of results. So as I said, the system's also responsive to BMP, but in a time-dependent way. So at day 0, this is 24 hours after plating in ROCK inhibitor, all cells are responsive to BMP, in this case by upregulating CDX2. It's most obvious in this image by all cells are red. In contrast, below that, 2 days later, when the epithelium is formed, it's only the cells in the periphery that are responsive to BMP. That is, you only see a red band of signal at the edge of the colony. So I'll show a short movie that demonstrates the self-organizing properties of pluripotent cells. In this movie, the images are updated every 2 minutes across a period of 2 days. In the first image, you'll see is as cells are landing right after passage. So this is acutely after passage. That video showed 2 days of development in culture after plating. These micrographs indicate the EPCAM signal before in the top red and after 2 days in the top right. I'm using different antibodies, so they have actually the same antibody but labeled with a different fluorophore, so they have different color, but the idea is that you see very strong localization in the top right of EPCAM to the edge. When we dissociate, you would get a heterogeneous population as shown on the right, where some cells are high red and other cells are low red. So we use image segmentation to identify these objects and count their signal, but also to produce complete lineage analysis to ask, is there a spontaneous organization, or do the high EPCAM cells find each other and organize based on pre-existing knowledge? Complete lineage analysis demonstrates that they find each other. So the argument for that assertion is that cells that begin high can give rise to both low EPCAM and high EPCAM cells after 2 days. In the schematic on the top left, cell A, this is an actual lineage a precise lineage of one cell. It divides twice to give rise to 3 daughters. One of them is at the edge and is shown in yellow, so it's a kind of high EPCAM cell, and there are 2 others that divide late in this time course, and they're both low. Similarly, cell B on the top left starts high and produces a range of signals. So if we are comfortable with the association between EPCAM and position in the colony, as I've shown in the movie and in these slides, then there's no relationship between the starting signal and the ending signal. Now, I've shown a schematic of about a dozen examples here where we see in the y-axis is the level of EPCAM signal, and in the x-axis is sort of just before the experiment starts and at the very end of the experiment. So there are only 2 time points here, but it shows you the change through time. And cell A that I had demonstrated in the top left is indicated with this red representation. So it starts in kind of a medium level, and there's 1 high daughter and 2 low. And cell B at the end is the highest one I could find in the field, and it gives rise to a range of signals at the end. So the point of all this analysis is that this system is unstable. That is, one begins to see specification into one lineage or another. That doesn't mean that's a one-way street. On passage, one can recreate pluripotency again. To summarize, this and many other studies we're doing in the lab, this concept of an epithelium is a structure that forms through time, where at the beginning of the culture paradigm, immediately after passage, all cells are in edge because they're not associated with other cells at relatively low density. As they divide and migrate, edges are formed spontaneously, as you saw in the movie. Those spontaneous changes are associated with an increase in the expression of proneural transcription factors and a decrease in the expression overall of the pluripotency factors POW, 5F1, OCT4, or NANOG. And as I'll address in the second part of the talk, there's a relative change in the level of PI3 kinase AKT signaling through time as well. Work that I'm not going to present here, but we're working intensely on in the lab is the association between these different developmental states and the formation of the mesendodermal lineage and the neuroectodermal lineage. So what I've described so far is the system, this concept of heterogeneity, where every cell, if you look at it in isolation, looks like the only cell like it in the dish. But when one puts developmental biology into the context, one has a much better understanding of what's going on. And what I'm going to discuss next is this concept of the neighborhood, a cellular neighborhood. That is, cells do not exist alone. They're influenced by their neighbors. And if one can understand the concept of heterogeneity, where it comes from, then you can capitalize on this knowledge and develop assays based on it. Liebherr Institute, we've made a significant investment in high-content imaging. The Alpharetta is our key acquisition system. We do all of our image management and analysis in Columbus, that is of two-dimensional images. For three-dimensional images, we would use Velocity. All of these data are stored locally on a high-performance server, and the processing is done there. But all of our, importantly, all of our multidimensional analysis and visualization is done with Spotfire. And you'll see the application of all these tools in the next slides. This is a typical image that one would see if you take a 3x3 array of images from a single well in a 96-well plate. And I represent these contiguous images as separate because most of us think and analyze images based on single cells. We have single cells as our target. What I'm going to discuss now is this concept of changing our perspective from high-content analysis to neighborhood content analysis, and really thinking about what is next to the cell that you're looking at. The way we address this problem is to capture an overlapping image matrix, to stitch them together into a single image, to find colonies, to find the colony edge, and then to map biological properties of single cells in Cartesian space to neighborhoods. That image is shown in the top left, and it's hard for your eyes, it's certainly hard for my eyes, to connect the dots and make a clear image, but that the DAPI channel from that same image is shown just next to it on the right. And when the objects are a little closer together, it's completely obvious that the edges are contiguous. We are focused on this ERB AKT pathway, and one of the best, most robust outputs from that is phosphorylation of AKT on serine 473. So the three micrographs you see, the edge finder and target nuclei, are shown, the red region of interest is expanded below. So I'm going to speak to the images below based on that region of interest, but you get a sense of where they come from in the greater context of the well. So serine 473 is seen here after stimulation as it's sort of localized to membranes. The edge finder algorithm will use nuclear segmentation to form a boundary that is defined here by the thin white line. So this thin white line is very important, and this is what I'm going to use as my to root all of the studies I'll discuss in the following slides. So the nuclei that are segmented are shown in outline on the far right. So with this analysis, what we're able to do is to look at association between position in a colony and developmental state or signaling response. So on the left is an original immunocytochemical stain. The green image in the middle is a representation of nuclei in this image on the left using just three columns of data. So the XY positions, and the third is a calculated distance from the edge, which is shown in green. So the more green the objects are rendered, the further away they are from the calculated edge. And on the right, the scatter plot shows that there's a clear association between NANOG, SOX21, and SOX2 expression. Here we're showing a fourth dimension, which is a very simple output from the high content analysis, and that's nuclear area in microns squared. So as nuclear area decreases, the expression of proneural transcription factors increase. And so it's very easy for us to represent all these dimensions in a single plot like this. So a high content assay incorporating these parameters is shown on the next two slides. So to set up the system, here what we're doing is stimulating with neuregulin 1 beta and neuregulin 1 alpha below. The consequence of stimulating iPS cells with these two ligands is completely clear. So with neuregulin 1 beta on the top, what you see is that the colony perimeter has a much stronger signal. And here we're measuring phosphorylation of AKT on serine 473. In contrast, below, the neuregulin 1 alpha shows elevated signal through time, and this is you're seeing a short time course. It's sort of 0, 5, 15, 30, and 60 minutes. But there's an elevation across the colony, so there's no dimensionality in the response with respect to the colony with neuregulin 1 alpha. And as I showed before with BMP, this neuregulin response with the output of AKT phosphorylation is also development and time dependent, not at the minutes but at the day level. So on day 2, there's no clear difference between the edge and the core of the colony, whereas 1 day later there's a huge contrast between the edge and the core. So we've incorporated these concepts into a much larger study that looks at multiple factors. Including in columns are the different ligands for the system, and in rows are the outputs. So this is the measurements in rows. And here we're incorporating 3 domains into each box of this large plot. The different colors reflect time after ligand addition, magenta being before addition, and then 5, 15, 30, and 60 minutes later. As with the other plots, the x-axis is distance from the edge, with the edge being leftmost, and the furthest objects from the edge are on the right, and the antibody signal is shown in the y-axis. So let's just look at the top, and you can see that the pan-AKT, so this is an antibody that recognizes AKT, whether it's phosphorylated or not, shows no obvious difference in signal through time or through space, that is in the x dimension, with the exception of the extreme edge, but all show a dip there. But there's no clear responsiveness in any domain to the pan-AKT antibody. Let's go to the third row, so way to the bottom. Another phosphorylation event on threonine 308 also shows, if you remember from the last slide, that much of the colony responds to some ligands. Here, all the cells are essentially at a high level and don't really change much with time. What I want you to focus on is simply the place where I have a red arrow. So this is the experiment I showed you earlier, where we add neuregulin 1 beta and measure serine phosphorylation on 473. And here, by 5 minutes, it's a huge elevation, only in the edge, not in the core. So there's a domain-selective response of this system at this time point. And what this has enabled us to do is to set up a screen for small molecules that perturb that signal in this domain. In conclusion, I've introduced the concept of heterogeneity in psychiatric research. The first point of heterogeneity is at the level of genes and people. The second is at the level of cellular heterogeneity, and the heterogeneity of cellular states within a colony. I provided a method for parsing heterogeneity into relatively homogeneous elements that can be analyzed in high content and high throughput assays. My intent in presenting this work is to extend our perspective on high content analysis beyond the single cell toward relationships between cells, but using existing technology that is currently applied to high content analysis. My coworkers in this project from the Liebert Institute are shown on the left and on the top right, and the acapella script that runs in server mode on Columbus was written by Ty Voss from PerkinElmer. He's now a close collaborator on this project. Thank you.
That was excellent. Thanks very much, Dan. I think our audience now has a great sense of the importance in developing high-content screening tools for the study of neurodevelopmental disorders. Before we get started with the Q&A session, I want to let everyone know this is your final chance to submit your questions for our speakers. We've gotten some really great questions already, but I'm encouraging the audience to keep them coming. All right, while the audience is submitting their final round of questions, let's begin the Q&A so that we can try to get to as many questions as possible. All right, guys, so let's get to the Q&A session. We'll get to the first question, and the first question is going to be for Satish. Satish, one of our audience members wants to know, what screening library did you use? How many CPDs? Did you confirm hits on multiple donors?
Yeah, we used a screening library from NCI, National Cancer Institute in the U.S., and it consists around 1,500 compounds. And we did confirm our hits on 9 different donors. So these were stem-like cells isolated from tumor samples from 9 different donors.
All right, thank you very much, Satish. Our next question is for Dan. Dan, how do other groups handle the analysis of signal in high heterogeneous cell-based assays?
Sure. Many people avoid these heterogeneous systems due to the low signal-to-noise ratio. Some groups have used fluorescent proteins that are driven by promoters with cell type specific expression, and one can then gate on that subpopulation. That's probably the best alternative solution to the sort of spatial analysis that I was describing.
All right, thank you very much, Dan. Satish, another question for you. Did you develop counter-assays producing negative results for VAC-Q? Is MOA specific to GBMs?
That's right. The mechanism of action is very specific and enriched in stem-like cells isolated from GBM. So the counter-assay to produce negative effects from vaccinol would be just to differentiate or make these stem-like cells lose their stemness, and we have done that. So when you take these stem-like cells from GBM tumors and add serum to it, they lose their pluripotency, and the effect of vaccinol also goes disappears on them.
All right, thank you very much for that one, Satish. Dan, is the acapella script for spatial analysis commercially available?
It is currently not, but as I understand from PerkinElmer, it will be one of the advanced building blocks that will be released in an upcoming version of Columbus and Harmony.
All right, thank you, Dan. Satish, one of our audience members wants to know, what is the PK characteristics of the D1 compound?
That study is underway, so we have not yet completed it, so you have to wait.
All right, well, that's a good answer. Dan, our next question is for you. How well do you think these cells model the cellular state in the nervous system of patients with psychiatric disorders such as schizophrenia?
Sure. Many groups attempt to identify phenotypes in mature neurons derived from iPS cells, such as my colleague here at the Liebert Institute, Brady Maher, or folks like Thomas Sudhoff, where the concept is that mature neurons are the most similar cell type to the cell most likely affected in psychiatric disorders. My intent in presenting this work and doing the type of studies we perform at the Lieber Institute is to build a model, not to model the adult nervous system, but rather to identify pathways that are differentially regulated and expressed in very early development, as many psychiatric disorders they show phenotypes late in adolescence, but have a very early developmental origin. The idea is that the genetic landscape that ultimately manifests in a clinical state late in adulthood is something that we may be able to tease apart using very early events in the development of the nervous system.
All right, thank you very much, Dan. Satish, one of our audience members, wants to know what instruments and software were used for the flow cytometry analysis.
We used BD Bioscience flow cytometry machine and FlowJo software for analysis of the fax data.
All right, thank you, Satish. Dan, which software did you use to do the segmentation?
All the segmentation that I presented was done with Columbus in server mode, so that's a PerkinElmer application for segmentation. All righty, thank you, Dan.
And Satish, this looks like the last question for Satish that we have. Satish, our audience member says that it was a very nice talk, and they would like to know what is the effect of D1 on the post-mitotic cells, such as neurons?
So we have not done that, but what I can say is we have done some behavioral studies on zebrafish, and we don't really see any big difference in behavior of zebrafish with their swimming pattern and socializing behavior. But though that study has not been done in vitro.
All right, thank you, Satish. And this looks like it's going to be our final question for the Q&A session for Dan. Dan, our audience member wants to know, can one recreate pluripotency again in this process?
Yes. My hope was to introduce this idea of instability, that is, the pluripotent state is maintained throughout this culture period, throughout 5 or 6 days in culture. But you start to see the emergence of expression of transcription factors that suggest lineage priming is going on. But the cells are still pluripotent, and I hope I made it clear both in the movie and in the associated analysis that pluripotency is never lost. We don't go beyond the point of no return, but that these evidence of lineage priming emerges through the first few days in culture, and then it's reset to a more primitive state. But all these cells are pluripotent at every one of these time points.
All right, thank you, Dan. And with that, we've come to the end of our webinar. So I'd like to remind everyone, again, that the webinar will be archived on our site for 6 months. That's www.genengnews.com. So if you missed parts of it and would like to watch it again, or you would like to forward it to your friends and colleagues, we always appreciate that. I'd like to thank Satish and Dan again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to PerkinElmer for supporting this webinar. So hopefully we'll see you again at another Gen webinar in the near future. So goodbye for now.